Hello, good evening. Hi, I know I'm late. I'm really sorry. There was a small technical glitch, so please bear with me. So I'm really sorry for the inconvenience caused, but anyway, welcome back. So we are here on our journey talking about the journey of life on earth, and that's biology. I'm really grateful and thank you for all those who have joined me. So good evening, Vishal. So I guess you're new, but I'm sorry for being late on your first class. Okay. So good evening. So good evening, everyone. So that's really nice. Okay. So today we are going to talk about very interesting topic, that is genetics. So genetics as such is interesting, but today it's all the more interesting because there's a pinch of more essence today. So what do I mean by a pinch of more essence? So we are on a NEET channel, so talking about genetic disorders. Okay, so which means you're all going to be doctors tomorrow. So obviously you'll be interested more about the application point of view, right? So theory is all fine, but you still want to know what kind of disease is there, what kind of treatment is there, etc. Right? So good evening, Harini. Good evening, Surendra. Okay, right. So this journey will be all the more interesting, and this will be the last lecture on Mendelian genetics. So with this, we'll be done with both your molecular genetics as well as your Mendelian genetics, right? So let's go forward and explore what are the diseases that are there. So we have not discovered all the diseases. So half the diseases we don't even know the name. Yes, so you're also new. So good evening, Mohammed. Hello. Okay, fine. So here I have few diseases, so we can't discuss all the diseases. So whatever is there in your NCERT, so whatever is applicable to you, so I have got those here. So we'll discuss only about those disorders. Okay. So before we actually go into Mendelian genetic disorders today, that is the last lecture of Mendelian genetics. So again, it's a gentle reminder. So you have a coupon code that is KR10. So you can use this on our eCareerPoint app. So you can use this coupon code to download or purchase any courses or products. So even if you're not going to purchase any courses or products, please download this app so that you can get my PPT and you can have a revision, okay? Because a lot of things are not on my slides, a lot of things I speak. Only hints or clues are there on my slides. So when you have to revise or when you have to recollect something, so make sure you have my PPT as well as your notes. And also making notes during the lecture keeps you focused. So anyway, I'm not going to teach for the entire 60 minutes ekdam. So I'm just going to give a break. So we are not going to talk for the entire 60 minutes. We're just going to talk for only 45 minutes, right? So make sure at least those 45 minutes you pay attention and you concentrate. Fine. And a gentle reminder, this coupon code is valid only till 30th of November. So make sure you use it before that. Okay, fine. So today, what are we going to talk? So what is the agenda for today? So that is what we're going to talk. So we're going to talk about mutation. So in the last lecture, we were talking about the chromosomal theory of inheritance, right? So we spoke a lot about genetic linkage, recombination, and we stopped with mutation. So today we will kickstart from there. So I will tell you what mutation is again. So there are various types of mutations. So how each mutation works. So what happens? So what is the logic behind that mutation? So what are the causes? What are the consequences of these mutations? So all that is what we're going to see today. So it is actually, you'll be surprised. Okay, things like this can happen. So we didn't create anything. So we are just studying the creation. So you'll also be amazed at how things can go wrong. So we are all fine today. We should all be really grateful because we are fine today. We are well and happy. So you can concentrate. You're normal. I am normal. It means your entire genetic machinery is way efficient, right? So now when we're talking about the genetic disorder, you understand how grateful you are, because if one mistake happens, I will show you what can be the consequence. Okay, so we'll kickstart from mutation, then we'll talk about pedigree analysis. So this is a very catchy term when you talk about genetics, right? So pedigree analysis, what actually happens, how it is studied, how it is analyzed. Now I'm sure every household, so at least one in a family has some problem, right? So no one is disease free. At least one of you, either you have some sinus, you have a cold right now, or your parents have your knee pain, anything, right? So none of us are disease free. So in this era, we're all going towards disease. It seems so, right? So what is this pedigree analysis? So someone at home might be diabetic. So you have your grandparents at home, right? So all of us are suffering from some disease. So this is just an overview. So how you can analyze, how you can predict. So all that is what we are going to talk. Then finally, we'll talk about genetic disorders. So genetic disorders are two types. One is Mendelian disorders and the other one is chromosomal disorder. Okay, so Vishal says a mutation is a change in DNA sequence. Yes, you're absolutely right. So that is what we're going to talk about in the next slide. Fine. So going on talking about mutation. So just like Vishal told us, I also have a definition for mutation. It is the sudden discontinuous variation in your genotype and phenotype of an organism. Okay, and it is due to the change in your chromosome or gene. So what do you mean by a genotype or a phenotype? So genotype is a genetic makeup of your body. Okay, the genetic makeup of your nucleus. So that is a genotype. So what is a phenotype? Phenotype is a physical observation. So phenotypically you're normal, which means your genotype is also normal. Okay. So when you look at a child, or when you look at a person who is physically challenged, or someone with a mental disorder, or someone with a genetic disorder, right? So their genotype will also reflect on their phenotype, right? So that is what I mean by mutation. So you can put it in simple term like Vishal told us. So any change in the DNA sequence, and this change can be very sudden, and it can be a very discontinuous variation. Okay. Yes, an observable physical property. Okay, so any observable change. So physically, when you observe a change, that is a phenotype. Okay, so remember, phenotype starts with P. So it means it correlates with your physical observation. Fine. So genotype, remember your genetic makeup. Okay, fine. Sorry. So along with recombination. So what does recombination give you? So when we were talking about the cell cycle and cell division, we saw that meiosis gives rise to recombination, right? So we spoke about that meiosis enables variations to happen, and variations are important for evolution. So along with recombination, even your mutations lead to variations, and thus it is also one of the important factor. So it's not that mutations are bad. Mutation has to happen, but it has to be a good mutation. For example, now we are currently in the crisis of corona, right? So we are in a pandemic right now. Still corona is not over, right? So there are chances that your coronavirus can also mutate. So when it mutates, there are high possibilities that it can become worse, it can become more harmful than what it is right now, or it can become less harmful, it can become less virulent. Okay, so mutations are good as well as mutations are bad. So mutation has to occur because it also gives way to variation, and variation is essential for evolution to happen. Fine, is that understood? Okay. So there are three types of mutations that we are going to discuss today. So the first type of mutation, and all the three types of mutations are depending upon the cause, like how it is being produced. So what causes that mutation? So based on the cause, we have three types of mutation. Fine? Okay, so the first one is called gene mutation. The second one is called chromosomal aberration. The third one is called as genomatic mutation. So we'll talk about each of the mutation in detail, so that you will understand what exactly are these mutations. So putting it in a simple term is easy. So any change in DNA sequence, so that change can also happen in three ways. So we are going to look at how these changes can happen. Okay, fine. First we'll talk about gene mutation. So what is actually gene mutation? So it is the alteration of DNA due to the change in the nucleotide sequence. So how is your DNA sequence? So your DNA is made up of base pairs, right? So your DNA is double stranded. So you have base pairs like ATGC, right? So if you have a change in one nucleotide, so that can be your gene mutation. So gene mutation, it occurs due to the change in the single base pair of DNA. So BP is base pair, a single base pair in DNA. What do I mean by base pair? Your DNA is double stranded, right? So this strand will complementary pair with the other strand. So what is the complement of A? T, for T it's A, G, C, right? So it is held together by hydrogen bonds. So if there is a change in the single base pair, then that leads to what is known as gene mutation. Fine? Okay. So one classic example of gene mutation is sickle cell anemia. Yes. So sickle cell anemia, we'll talk about it in detail. It is one of the disorders that we're going to talk about. For now, remember, sickle cell anemia has a change in a single base pair. Okay? Fine? Sorry, so which means you have to remember sickle cell anemia has a point mutation. It does not have a major change, just a small change in a single base, and that creates all the chaos. Fine? Okay? So going forward, we are still in gene mutation. So gene mutation, how it occurs. So what causes that small change to occur? So what causes that point mutation to occur? So first one is your frame shift mutation. So either deletion can happen, okay, or insertion or addition can happen. So for example, when I say this is your double-stranded DNA, okay, so I have sequence like ATGC. So here you have TACG, okay. So when I say insertion or deletion, if I mean deletion, so this sequence is lost. So this TA base pairing is lost, okay. Again here, this is lost. So that is deletion. Similarly, when I say insertion, a new sequence is added here. So when I add a new sequence, remember your amino acids are read in triplets, right? So it is 3 codon. So this will again change the proteins that it is coding for, right? So that is the major problem. The second reason is substitution. So you have replacement of one base by the other. Everything is fine, but here C is replaced by G. So again here it changes. So now this changes your reading frame, right? So that is the problem here. So your substitution can be of two types. So one is transition and one is transversion. So repeating again, your DNA is made up of nitrogen bases. Your nitrogen bases are of two types. One is purine and the other one is pyrimidine, right? So purines are your adenine and guanine, okay? And your pyrimidines are thymidine and cytosine, right? So when a purine gets exchanged or substituted with a purine, when A is replaced or substituted with G, it is called as transition. But when an A is substituted with T and a G is substituted with C, when a purine is substituted with a pyrimidine, it is called as transversion. Fine? Sorry. Okay, so look at the point mutation. So this is your normal sequence. Okay, so this is completely normal. This is how a normal a gene will code for an amino acid. Okay, so I can. Okay, so now if there is a point mutation, understand this single base is changed here. Right? So if the single base is changed here, TGG codes for tryptophan, but TCG codes for serine. So this changes the entire makeup, right? And hence all the codons get changed. Okay, so it changes your reading frame. So it is getting inserted here. Okay, so it is not getting substituted, it's getting inserted. So if you insert a C here, if a new base pair is inserted, so it changes your entire reading frame, meaning your entire gene codes for a different protein or an unwanted protein. Hence it will be reflected on your phenotype. So this is what mutation is all about. So this is specifically gene mutation. Okay. Now, next type of mutation, so it is chromosomal aberration. So from the name, it should be clear what it means. So any change or any mismatch in the chromosome causes chromosomal aberration. So this is also one of the major drawbacks. Okay, so as long as you are fine, you won't know the consequences of that, right? So people say only when you get your toothache, when you get your fever, you will know the pain. It is the same way. So to study, it is fine. But when you start looking at the consequences, when you start treating the patients of that disorders, so it is really painful. But let's see how it happens. So chromosomal aberrations, it is the loss or gain of a segment of DNA. So when an entire segment of DNA is either lost or a new segment of DNA is inserted, so that results in the alteration in the chromosome, because all of us know the normal equilibrium, right? So suddenly, so now I tell you tomorrow I have a class. Okay, so tomorrow we are going to have a class at 4 p.m. You will all be prepared for the class. Now if I suddenly keep a surprise test, you will not know how to react, right? So half of you will be frustrated with me, half of you will be angry with me. So sudden equilibrium gets changed. So similarly, imagine if an entire new segment of DNA is lost or gained, so your genetic machinery is clueless. So then it does not know what to do, it just proceeds casually, okay, and making a lot of mistakes. So that is the alteration in chromosome. Fine. So it is commonly observed in case of cancer cells, okay. So what are the important types of aberration? One is deletion and the other one is duplication. So a segment of DNA, when it is deleted, so you miss out a particular protein that it is coding. For example, you have 3 genes, A, B, C. Suddenly B gene has been deleted due to mutation. So your protein that is producing, so the gene B that has to produce a protein, that is being lost. So that function is being lost, okay. So that will lead to chromosomal aberrations. So your phenotypically you will be affected because of the loss of that protein. Fine. So then it is duplication. So when you have twice the amount of B gene, so you have A gene, B gene, again a C gene. So you'll have twice the amount of protein produced. Again, that is also not needed, right? So everything has to be in the right amount. So when you have twice the amount of protein produced, again, that is a disorder, okay? So that happens due to duplication. So your deletion can be of 2 types. One is terminal and the other one is intercalary or interstitial. So just like the name says terminal, if the terminal portion of your chromosome is deleted, so that is also a chromosomal aberration. Okay, and interstitial portion, if the in-between portion is deleted or lost, so that is also a type of deletion. So talking about duplication, when you have addition of a segment, so you have a gene repeated twice, okay. For example, it can be a single gene that is duplicated or a gene set that is being duplicated. So I have an example. So this is the normal chromosome, okay. So it has the terminal portion perfectly, everything is there, okay. How duplication takes place, okay, I will tell you. So now you have to, when you have deletion happening, so this terminal portion is deleted, so this is terminal deletion. When you have an interstitial portion deleted, so this is your intercalary or your interstitial deletion, fine. So how duplication happens? For example, let me do here. I have a gene, so this is the normal gene set A, B, C, fine, okay. Now, what will happen in case of duplication is now I have A, so I'll have twice the set of B genes. So your B gene has been duplicated. If you ask me how duplication happens, you have something called as transposons. It is out of context because it's not there in your NCERT. But since if you're very interested and you want to know the logic behind, you have something called as transposons. So transposons are basically jumping genes. They can cut, copy and paste, okay? So sometimes you have genes in your DNA which can code for your transposons. Okay, so you have genes which code for transposons. So when these transposons are coded, what happens is they act on a particular gene, they copy paste them. So as a result, you have duplicated genes. So this is also harmful and this is also a type of mutation. Fine? Okay. So the last type of mutation is your genomatic mutation. Fine? So what is a genomatic mutation? So the change in chromosome number. So this is the last type of mutation and it means a change in the chromosomal number. So for example, you and I, we have 46 chromosomes, right? So that is 23 pairs of chromosome. So if you have a change in the chromosome number, so what if you have 22 pairs? What if you have 45 chromosomes? So that is called a genomatic mutation, okay? So you have visible effects on the phenotype. Fine. So here you have two types under genomatic mutation. One is aneuploidy and the other one is euploidy, okay? So what is aneuploidy and what is euploidy? So we are going to talk about that. So aneuploidy is due to the non-disjunction of the two chromosomes of a homologous pair. So this terms might be complicated. What is non-disjunction of chromosome of homologous pair? So every term looks complicated, right? But it's nothing. So when we were talking about meiosis, so you have crossing over happening, right? So when there is no proper separation of your homologous chromosome. So at the time of crossing over, so you have bivalence like this. So the information has to be crossed over, right? So this gives you recombinant one. So once crossing over has been done, so this has to be separated. When there is no proper separation, when there is no proper disjunction, so that leads to aneuploidy. Fine? So that is called as aneuploidy. So your examples are trisomy, Down syndrome, Klinefelter's syndrome. So we'll talk about all this syndrome very soon. Okay? So trisomy, you will have you have one extra chromosome. Okay? So for example, you will have 3 copies of 21 chromosome. So you ideally have 2 copies, one from your father, one from your mother. But you have one extra copy, so that is trisomy. Okay? So that is trisomy, that is Klinefelter's syndrome. So we will discuss about all this. And Turner's syndrome, you will have only 45 chromosomes. You will have one less. Okay? Fine? So that is Turner's syndrome. Okay? So look at this. This is disjunction. So in a normal disjunction, what has to happen? This has to come here. This has to come here. Fine. After this, what happens? Ideally, there should be 2 sets coming, but there are 3 sets of chromosomes coming. So this is because of mutation. So you have 3 sets of chromosomes. This is trisomic. You have only 1 chromosome instead of 2 sets. So that is monosomic, right? So this is how your genomatic mutation happens. Fine, is this understood? You have a change in the number of chromosomes that is coming. So ideally you should have a pair. So one from your father, one from your mother. But if you have extra, or if you have lost one pair, so that is called as aneuploidy. So now what is euploidy? Okay, so this is also a type of genomatic mutation. So it is again the change in number of chromosomes, but it is a multiple number of chromosomes. So in aneuploidy, you can either have loss or gain, but in euploidy, you only have double the amount. Okay, so you never run short here. You have twice the amount. You might have 92 chromosomes, something like that. Okay, but euploidy is commonly observed in plants. It is not observed in humans. Okay, so due to variation in one or more haploid set of chromosomes. So at the time of meiosis, what happens? Your mother cell divides to give 2 haploid cells. But if there is a fault here, so one cell is 2n, the other one is n. So such a condition gives rise to euploid. Fine. So again, euploidy is of 2 types. First one is haploidy and the other one is polyploidy. So haploidy means only one set of chromosome is extra. So instead of 23, instead of 46 chromosomes, you have 47 chromosomes. But polyploidy is when you have twice or thrice the amount. Okay. So that is about the mutations. Now we are entering into genetic disorders. So when I talk about genetic disorders, the first thing you need to understand is what is pedigree analysis. Because with the help of pedigree analysis only, we'll be able to analyze, okay, so how is the gene being passed on. So if you have a defective gene, so from which lineage it is being passed on. So that helps us with understanding more about genetic disorders, and that is your pedigree analysis. Fine. So these disorders, so genetic disorders are definitely inherited. Okay. So in pedigree analysis, so the inheritance of a particular trait is represented over a family tree generation. For example, it is said that if a father has cancer, sometimes the cancer can be hereditary and it can be passed on to the daughter. Okay. For example, if the mother has cancer, you have high chances that it can be hereditary and it can be passed on to the sons. So how do you study this? For example, the parents do not have, but the grandparents have and the grandchildren are affected. So there's something being passed on. So in the parental generation, your gene is silenced. So you need to trace back the origin, right? So that is with the help of pedigree. Okay. So it's all in the family. So everything, every disease that you have, sometimes everything is inherited. Okay. Not from your immediate parents, but from your grandparents or your great grandparents. Okay. So for your pedigree analysis, you have certain tools here. Okay, so just like that, you can't do analysis. You need to have some kind of tools to do something, right? So when you have a square box, it is represented as a male. Okay, so why I am teaching this is when you have genetics topic, sorry, so in NEET, so you will have questions like you will be given a pedigree analysis and you will be asked to find what is the probability of the generation having a disease. For example, I'll show you a pedigree in the next slide. So they will just give you a pedigree chart and ask you what is the probability that in the third generation the son will have a disease. So for that you should know how to read a pedigree analysis, right? You should know how to analyze a pedigree chart. So for that you should know what are the tools, fine? So you have a squared box, so that represents your male. Then you have a female, so female is circle, okay? So when it is in between, so you do not know the sex, okay? So sex is unspecified here. So when it is, so this can be in various colors, but here I have used this combination. So I am choosing this as your reference. So when it is in red, it means that they are affected individuals, they are affected with a particular disease, okay? Then male and female, this is mating. When you have a double line, it is consanguineous mating. So what do I mean by consanguineous mating? So when you marry within relatives, right? So for example, if you marry within relatives, your ancestors are the same. So the same affected gene can be passed, right? So that is why these days you have genetic counselings happening in hospitals, and they strongly recommend not to have consanguineous marriages because the probability of the genetic disorder spreading is really high. Okay, so this is consanguineous marriage, fine? So this is a father, mother, and they have two child. So this is again a girl child, this is again a boy child, fine? So this is the first child, this is the second child, okay? Then here it means that the child is affected, okay? And finally, you have 5 unaffected offsprings. So when it is a diagonal or when it is a diamond shape, it means it is unaffected here, okay? Okay, so with this introduction, I go into pedigree. So this is how a pedigree chart looks like. So now, don't look at what is written below, just look at the pedigree chart. So what is this? So this is the mother, so this is the female, right? So this is obviously the mother, so this is the father, right? So the father is not affected, your father is normal. Your mother is affected because it is in red, right? Now, talking about their children, so out of the 5 children, so 2 kids are affected, right? Again, their further generation is also affected. So this is how a typical pedigree chart looks like. Now, what you have to understand here is the children are affected because the parent is affected. One of the parent is affected. Since one of the parent is affected, the child is affected, so it is autosomal dominant. Since the parental generation is affected, the child is affected, so this is a dominant disease. Okay, so figure A represents autosomal dominant. Autosome means your chromosomes other than sex chromosomes, right? So you have autosomes plus your sex chromosomes. So autosomes is other than your sex chromosome. You have a mutation in your autosome, and that is directly coming from your parents. So it is autosomal. Fine. Now come to this pedigree analysis. Here both the parents are normal, but how come the child is affected? Which means they are normal, they are not affected, but they can be carriers. So they can have only carriers, so it does not affect them. But when it is being passed on to the next generation, it affects their kids. So such a kind of disease, such a kind of linkage is called as recessive, autosomal recessive. So again, this is on your autosome, the mutation is on your autosome, but it is recessive. It is not expressed in your parental generation, but it is expressed in your offspring generation. Fine. So this is how a typical pedigree chart looks like. Okay. So going further, we are going to talk about genetic disorders. The first one, genetic disorders are divided into two types. One is Mendelian disorders. So what does Mendelian disorder mean? So when you have a change in your DNA sequence, that is your Mendelian disorder, okay? So a change in a, so when I say there's a point mutation, for example, your sickle cell anemia has a point mutation. So that will come under your Mendelian disorders. The next part is your chromosomal disorder, when you have a shift or change or loss or gain of a particular set of chromosome. So that comes under your chromosomal disorder, fine? Okay. So your color blindness, hemophilia, sickle cell anemia, phenylketonuria. So all this comes under your Mendelian disorders. So what comes under your chromosomal disorders? Your Down syndrome, because here you have an extra set of chromosome or you are in short of a particular set of chromosome, right? So we'll talk about all these diseases in detail. So with that we'll be done, okay? So don't worry, I'm not going to complicate. I'm just going to give you an idea what happens in each of the disease. So where is the fault? For example, color blindness, it might be self-explanatory, but in case of sickle cell anemia and phenylketonuria, you should know what is being mutated, right? So that is my logic. So let's see. First one is color blindness. So how many of you can differentiate between red, green and blue color? Specifically red, maroon and orange. So if you can properly distinguish between red, orange and maroon, it means you are normal. You do not have color blindness. Okay, that is on a funny note. But still, let's see what is color blindness. It is a sex-linked recessive disorder. So this is a very important statement, sex-linked recessive disorder. When I say it is sex-linked, you have a mutation on your sex chromosomes only. It is not autosomal, it is sex-linked. So you have your mutation on your sex chromosome, okay, mostly on the X chromosome. Okay, so whenever I say sex-linked, the mutation is on your X chromosome only, because your female has XX and your male has XY, right? So from this, either one of the X has to come. Here, it will be either X or Y, right? So mostly your mutation happens on your X chromosome, and that is sex-linked. So what do you mean by recessive? When the parental generation are only carriers and they are not affected. Okay, so that is sex-linked recessive disorder. Fine. So it is a defect to distinguish between your red, green cone of your eye. So what happens? You have three types of cones, okay? So depending upon the particular wavelength, a particular cone gets activated in your eye. So when, so at a time when you're perceiving so many signals, it is a mixture of all these stimuli. So for example, if a particular cone is not able to observe this visible wavelength, then you won't be able to see that color, okay? So a lot of people have a confusion between red, orange and maroon. Suddenly it looks like everything looks like red to them, or everything looks like orange to them. So that close color differentiation is what is color blindness, okay? Sometimes people cannot see between green and red also. So can you differentiate how many colors are there here? So if you can differentiate how many colors are there here, then it means you're normal. Okay, so it might not be clear from such a distance, but then let us zoom it and tell me. Okay, so where the mutation happens? The mutation particularly happens on genes present in your X chromosome. Fine. Okay, so that is about color blindness. So going forward, we are talking about hemophilia. So what is hemophilia? So it is again sex-linked recessive disorder. So I have explained this term. So this is important. Okay, so again your mutation is on your sex chromosome. It is transmission from unaffected carrier female to some of the male progeny. So here your female is the carrier. So when I say recessive, the parental generation are not affected, but they are carriers. Okay, so mostly the female are the carrier. So when the mother is the carrier, there are high possibility that the son is affected, because if mother is XX, okay, and the son will be XY, and this X comes from the mother, right? So whenever the mother is the carrier, it is always the son who is affected. Fine. Okay. So proteins, so what affects in case of hemophilia is that you will have proteins that are important for blood clot, so that will be majorly affected. So what happens in case of blood clot? So look at this pathway. So what is blood clot? So when you get hurt, you start bleeding, right? So your blood starts oozing out, but it's not safe, right? So you can't lose all your blood. So there has to be some mechanism to stop it. And brilliantly, your body has its own mechanism, and that is called as the clotting mechanism, right? So if your blood does not clot, it is highly dangerous. So what happens in the clotting pathway? It is a very huge pathway. We are not going to talk in detail about each of the pathway, okay? I will just explain in a rough way so that you understand what is this pathway. So you have a protein called prothrombin. Prothrombin, in the presence of certain factors, will get converted into thrombin. So prothrombin is an inactive version and thrombin is an active version, okay? Now your thrombin will activate fibrinogen and it will become fibrin. And this fibrin is responsible for making your clot happen, okay? So this fibrin is important. So if your fibrin is important, what is important for the activation? That is thrombin. For activation of thrombin, you have so many factors. So you have factor 12, which is inactive, then it is converted into factor 12A, which means it is activated, okay? 12 activation. Again, 11 has to be activated, 9 has to be activated, factor 10 has to be activated. So only when factor 10 is activated, your thrombin can be converted to, prothrombin can be converted to thrombin, right? Okay? So, for this to be converted to this, a lot of factors play a role. If there is a mutation in any one particular factor, this process will not happen. So, your entire clotting process is completely halted, right? So, if there is a mutation, for example, if factor 9 or factor 10 is not produced, if it's not activated, your entire coagulation pathway will not be activated, right? So, that is the major problem in case of hemophilia. Fine. So this is what happens in case of hemophilia. So you'll have non-stop bleeding because your clotting mechanism is halted. So all of you know Queen Victoria, right? She was a carrier for hemophilia. So all the male in her entire family, they are affected with hemophilia. Hemophilia is pretty common these days. Okay, so we don't have a majority of treatment. So what you can do, if you have a mutation in a particular gene, you artificially inject that factor so that you have the process of clotting happening. Okay, fine. So that is about hemophilia. Okay, so Vishal, thank you. So now next disease that we're going to talk about is sickle cell anemia. So what is sickle cell anemia? I told you it is a classic example of point mutation. But what is sickle cell anemia? It is an autosome linked recessive trait. So this is not on your sex chromosome. So the mutation is on your autosomes, right? And it's again recessive. Fine. So when both the parents are carriers for the genes, so the parents are not affected, but they carry the mutated gene. But when it is passed on to the offspring, it becomes dominant and it is expressed. Fine. So you have alleles, right? So we spoke about alleles. So you have two things. So your parents will have one normal gene and one mutated gene because they are carrier of your mutated gene. But when your child has both the mutated gene and it shows the effect, your child will be affected. So your disease phenotype is HBS, HBS. So what is HB? HB is hemoglobin because sickle cell anemia acts on your RBC. Okay, fine. So what happens in this mutation? So GAG codes for glutamic acid, but instead of G, it is changed to U. Now U codes for valine. So it is a completely different amino acid. So when it is a completely different amino acid, the end function of the product will also be different, right? So that is what happens in case of sickle cell anemia. In your normal blood cell, when your glutamic acid is coded, so it produces a normal blood section, right? So you have the proper carrying opportunity for your hemoglobin. But when you have this mutation from glutamic acid to valine, your RBC is in the shape of a sickle. So that is why it is called sickle cell anemia. So when it is in the shape, it is not efficient enough to carry hemoglobin forward. Okay, so that is the whole logic of sickle cell anemia. Fine? Okay. The next disease is phenylketonuria. So we have already discussed about this disease. So let's again revise. So it is an inborn error of metabolism. So basically there's no external mutation, but it is an inborn error. So what happens is it is also inherited as autosomal recessive trait. So autosomal, again, it is not sex linked and it is recessive. So the parents are carriers here. Okay. So here what is mutated is an enzyme. So the mutation happens on the gene coding for an enzyme. So what is that enzyme? So it is an enzyme that converts phenylalanine to tyrosine. So according to your cellular metabolic pathway, your phenylalanine is an amino acid and it has to be converted to tyrosine. So for this conversion, for all your cellular or metabolic process, for the conversion you have enzymes, okay. So every cellular process, every particular step in your cellular process, it is aided by an enzyme. For example, the enzyme that converts this, the enzyme that aids this reaction is lost. So this reaction will not proceed. So you will have a lot of accumulation of phenylalanine, right? So you will have accumulation in the form of phenylpuric acid. So that is bad. It will also get accumulated in various parts. So not letting proper functioning happen. For example, in severe cases when it is not detected, it will lead to mental retardation as well. Okay. So this is how people with phenylketonuria are diagnosed. So what treatment can you do? You can inject this normal enzyme, but how often will you do? Your metabolic processes are happening every day. So you try to change the mutation in the gene. So when I say there is a mutation, for example, let's take the example of sickle cell anemia. So the mutation is from GAG to GUG, right? So every now and then you can't keep injecting, right? So you would like to change the gene. So how will you change the gene? How will you correct the repair? It is not that you will take the gene out from your body and take out gene and put you. It is easier said than done. It's not possible, right? So your DNA itself is microscopic. And how will you look at the basis that is present? So it is not possible. So gene therapy is a more tedious process. There is gene therapy and there are treatments for this, but the success rate is very, very less because it has to be done with a lot of care and a lot of precision so that there's nothing that goes wrong. Okay. So you're dealing with someone at the genetic level and it is not easy. Fine. Okay. So the next is thalassemia. So what happens in case of thalassemia? So thalassemia is again autosome. So the mutation is on your autosome, not on your sex chromosome. It is again recessive. So your parents are carriers. So the disease is transmitted from your parents. So here it is either mutation or deletion. So you have a particular deletion in that gene that codes for a protein. So what is that protein? So thalassemia basically is found in the Mediterranean population. It was initially found in the Mediterranean population. So you have two globulin chains, alpha globulin chain and beta globulin chain, okay, of your hemoglobin. So when you have mutation in alpha globulin, you can also have mutation in your beta globulin. Fine. So the thing is HbA1, HbA2, okay. So this is your alpha chain. So this is on chromosome 16. So you can have mutation either on chromosome 16. So your alpha globulin will be affected. So when you have mutation on HBB, that is the beta globulin chain on chromosome 11, your beta chain will be affected. Fine. So this is normal, but in case of thalassemia, it is not like your normal, right? So it is not concave shape. So the ability to carry your hemoglobin will be less. So this is the difference between thalassemia and what was the other thing that we saw, sickle cell anemia. Fine. Okay. So the mutation is different, but both act on your RBC. Okay. Okay. Now coming to chromosomal disorders. So long we have been talking about Mendelian disorders. So you saw how a single change in your DNA can lead to such disastrous change, disastrous consequence. Okay, that is fine. Okay, so you have small change and yet it gives you a lot of consequence. So now let's see what happens if you have an extra pair of chromosome or if you are in need of one pair of chromosome, only if you have one set of chromosomes. So that is chromosomal disorders. Fine. So absence or abnormal arrangement of one or more chromosome is your chromosomal disorder. Fine. So aneuploidy. So aneuploidy, where did we read? In your genomatic mutation, right? So aneuploidy is either loss of gain of chromosomes. So we are going to talk about each of this syndrome. Okay. So polyploidy, it increases, it is the increase in the whole set of chromosome in an organism. So mostly polyploidy is observed in plants. Fine. So trisomy is also there, monosomy is also there. So trisomy when you have extra set of chromosome, monosomy is when you have only one set of sex chromosome. Okay, any particular chromosome, fine, it need not be sex chromosome. So this is your normal chromosome, but in case of your chromosome with deletion, so this portion is deleted. When this portion is deleted, it might have essential genes that it codes for, right? So that is being lost. Okay. So now let's look into first Down syndrome. So what is Down syndrome? So it is the additional copy of chromosome 21. So you have 23 chromosomes. Okay. So you remember what is karyotype, right? So I showed you the picture of karyotype when we were talking about cell, the unit of life. So what was the karyotype? It is the representation of your chromosomes. So you study the morphology of chromosomes, right? So if you remember the picture that I showed you, not all chromosomes were of the same size and shape, right? So each chromosome was of particular shape and size. Your sex chromosomes were of the smallest size. Your X chromosome is the smallest, right? So each chromosome has a particular shape and size. So if you look into that, so if you have to find a chromosomal abnormality, if you have to find a chromosomal disorder, you do karyotyping, and you find, so you have a normal person's karyotyping and you compare it with a deceased person. Okay, which chromosome is affected, which chromosome is mutated. Okay, so that is what you do. So here in case of Down syndrome, you have an additional copy of chromosome 21. So additionally, so ideally you should have one pair from your mother, one pair from your father. But if you have one extra, that becomes Down syndrome. This is especially on chromosome 21. So why does this happen? So your normal sperm, when it fuses with an abnormal egg, so during the process of egg production, so that oogenesis, so there is no proper separation. So as a result of the abnormal separation, you will have 2 copies of 21 chromosomes in your egg. So when your sperm fuses with this abnormal egg, it will produce additional copy of chromosome 21. So this is called as Down syndrome. Fine. So this was first described by Langdon Down. So hence, in honor of his name, we kept it as Down syndrome. So it's the name of a person. So next time when you write, ensure D's caps. Okay. So that is how you write a person's name. So it is a fusion of a normal sperm with an abnormal egg. So the egg will have an extra copy of 21 chromosome, and hence when it fuses with your normal sperm, it produces an additional copy. Okay. So basically it is due to chromosomal non-disjunction. Okay. So small round head, partially open mouth. So these are the symptoms of your Down syndrome. Okay. So this is how the baby will look like. Okay. So basically when this happens is when a woman with more age, so above 44, 45, 40 years of age, so when the ability of the chromosome to divide properly is lost. So that is when cases like this happens. Okay. So Klinefelter's and Turner's syndrome. So Klinefelter's is when you have an additional copy of X chromosome. So you will have 2 copies of X chromosome. So that makes it 47 chromosomes. Okay. So overall masculine development will be there because XY is there, so it will have a masculine development. Since it has 2 X chromosomes, it will have a feminine development as well. That is, it also express the production of breast will be there, so that is called as gynecomastia. Okay, so you will have both masculine and feminine development. It will be an overall masculine development, but some features of your female will also be there. Okay, so these kind of individuals, they are sterile. They won't be able to produce a fertile gamete. Fine. Then tuners, so it is absence of 1 X chromosome. So here you have only 45 chromosomes and not 46 chromosomes. Fine. So such females are also mostly sterile. So this is about your chromosomal disorder. Yes, so this is your karyotyping that I want to show you. Okay, so if you look at all the 23 sets of chromosomes that is shown here, not all chromosomes are of the same shape and size, right? So everything is different. So look at this chromosome 15, look at chromosome 21. So chromosome 21 is the smallest. So look at the size, look at the chromosome Y. So this is how you identify a chromosomal disorder. You do a karyotyping, okay, and then you find out which chromosome pair is extra, which set is extra. Fine. So here you have 2X. So this is the karyotype of your Klinefelter's syndrome. Okay, fine. Okay, so this is all about your genetic disorders that I want to talk to you. So with this, we are done with your Mendelian genetics. So quickly revising or recapitulating what we spoke for the last 1 hour. So I have 3 questions. So if you can answer, so that is a quick revision. So color blindness is what kind of disease? Is it a sex-linked or autosomal recessive? So that is what I want. So quickly going back again and revising your color blindness. So what did we talk about in color blindness? So whether it is autosomal dominant or recessive, do you remember? So color blindness was the first disease, Mendelian disease that we spoke about, right? So what did we talk about? This is important. So you will have questions like this. Color blindness, you should know. Sometimes they will ask you the end result and then your answer will be to find out whether what kind of disease it is, whether it is dominant, recessive, whether it is sex-linked or autosomal dominant. Okay, so here your color blindness is sex-linked recessive disorder. When I say sex-linked, so it is on your sex chromosome, mutation is on your sex chromosome. When I say recessive, it means your parental generations are just carriers, they are not affected, but your offspring generation will be affected, fine? So now going forward, so each disease you have to remember whether it is sex-linked, recessive, autosomal dominant or recessive. So that is more important. So when you study, make a tabular column and then study. So that will give you an idea how it is being affected in a pedigree analysis, okay? So maybe in one class we will solve all the questions of genetics, all the previous year questions of genetics. So you will know how to read, analyze a pedigree chart, okay? So quickly going forward, so I just have not much questions, just 3 questions to summarize what we spoke, so that you should know what kind of questions comes into picture, fine? Okay, so here we are. A gene that shows its effect on more than one character. So this we didn't talk today, we spoke in the last class. A gene which can show its effect on more than one character. So one example that I was talking in the last class was your skin color, right? So you have an intermediate, dominant one will have a dark color, then your recessive one will have a light color, then you have an intermediate. So that term is called as pleiotropic. Okay, so the entire chapter I am taking the summary, okay, not only of this lecture. So you should also know the previous lecture before answering it. So a gene that shows its effect on more than one character. No, it is not polygenic inheritance. So I am talking about a particular gene. So polygenic inheritance is different. Here when a gene that shows its effect on more than one character, that is pleiotropic, fine. So the last question. So the recessive genes located on X chromosome in humans are always. So when I say recessive genes, it means the mutated genes that I'm talking about. So we saw a lot of disease that I said is sex-linked recessive, right? So those recessive genes that are located on X chromosomes in humans are always expressed in males, which means your female is the carrier. In the next generation, they're always expressed in males. Okay, so that is what I want to do. I didn't give you the options because it is just a summary. So we'll take another whole lecture to solve all the previous year questions in genetics, because according to class 12 syllabus, genetics has high weightage, right? So we'll take one class to solve all the important questions. So we'll solve a lot of pedigree analysis, a lot of other questions related to your chromosomes and the packaging of chromosomes and Mendel's crosses as well. Okay, so this is all about Mendelian genetics. So today we are done with Mendelian genetics. So wait till you get the notification of my next thumbnail. Okay, so again we'll come back with a high weightage chapter because we're talking about class 12. So again, it's a gentle reminder, I teach for class 12 on Monday, Wednesday and Friday, and for class 11 on the remaining 3 days, that is Tuesday, Thursday and Saturday. Fine? Okay, so if you really like the session, if you understood something out of it, if you're genuinely happy, so please do like, share and subscribe. So please support us on this, because it will ensure that we have a broader reach, and students who do not have the opportunity to pay for a class will know that they have a free opportunity, they have such a beautiful opportunity available online, because net is there with everyone, right? But they should know that something like this is available. So opportunity will wait, sometimes it will knock, but always it will not knock. So please help us in carrying this forward. So please do like, share and subscribe. So with that, I am signing off. So tomorrow join me at 4 p.m. So we'll be talking about a class 11 subject topic. So it is again a good chapter. So that is biological classification we'll be talking about. So when I say biological classification, all of you are scared. But don't worry, we'll make it all the more interesting. So join me tomorrow at 4 p.m. for biological classification. Okay. So until then, this is me signing off. So thank you all those who joined me. Bye.